we jump forward here, couple things. First, couple things. So we're starting on the nervous system today, and so that's going to be our next kind of block of material. In 2 weeks from today, we're going to have our third and final lab practical. So that'll be uploaded onto Canvas, and I'll give more information about that as things get closer. But it's going to run essentially the same way as lab practical 2 went. And then 3 weeks from today, we will have exam 4. So that will be where we'll have, you know, the last material for the nervous system block. Okay. So I'll give more information on that as we get closer. But like I said, today we're going to be starting on the nervous system. So is everyone able to see the PowerPoint slide?
Yes.
Yes, sir.
Yeah, okay, perfect. Okay. Okay, so today we're going over the nervous system. So before we start with the nervous system, what do we know already about it?
Well,
So there's 2 branches of nervous system, central and peripheral nervous system.
Okay, so we got the central
and
and peripheral nervous system.
Mhm.
Peripheral.
Um,
Okay. What else?
Correct me if I'm wrong, but isn't there sympathetic and parasympathetic? Or no, is that
Yes,
something?
we have sympathetic. Yes, absolutely. So we got the parasympathetic and sympathetic. Do you remember which one does what?
Flight is sympathetic and parasympathetic is the opposite, right?
Yeah. So sympathetic is your fight or flight,
Uh-huh.
and then the parasympathetic is your rest and digest.
Resting.
And so we'll talk about that a little more later today, and then in a couple of weeks as well. Cool. What else do we know about the nervous system?
Isn't it like the whole nervous system is like spread around the whole body? It's like nerves itself. Like they're all around, aren't they? Right?
Yeah. Absolutely. Yeah. So the nervous system is going to be all over the body. Perfect.
Oh, wait. I think I have something to add. Does nervous system, wait, I don't think nervous system has like the ability to heal like fully, right? Like it can't fully heal. Is that correct? Right?
Yes.
All right.
Yes, absolutely. So we'll talk about that in a little bit more detail today as well. So it can't regenerate.
Yeah.
Or it can't regenerate well. In some cases, can't regenerate well. In other cases, it can't regenerate at all.
Yeah.
Cool. Perfect. What else do we know?
It
See if we can get
uses
one more.
sensory receptors to gather via information stuff. Like
Yes. So there's going to be lots of sensory receptors, and they're going to be able to pick up on lots of information, right? Pick up on lots of information both in our external environment as well as in our internal environment as well. Perfect. Cool. So we know quite a bit about the nervous system. One thing I forgot to mention is I forgot to upload these before class today. The PowerPoint is now uploaded. So if you weren't able to get to it before, it's now available on Canvas. So you should be able to get to that now. But as we can see, the nervous system has a lot of different jobs, right? It's going to have 2 different components, or 2 different kind of overall components, the central and the peripheral nervous system. And it's going to be all over the body, and it's going to be able to regulate lots of different things that are going on throughout the body. And so we're going to be taking a look at what it's able to do kind of in a more general perspective today. And then Wednesday, we'll look at these spinal nerves and how the spinal cord is built and what it can do. And then we'll also look next week at the brain and see what it's able to do as well. So we're going to be getting a good, pretty in-depth look at the nervous system going forward here. So our game plan for today is to take a look at how the nervous system is organized and how it's structured, the different functions that it's going to be able to perform. We're going to be looking at the different cells that are going to be responsible for allowing the nervous system to work the way it needs to. Looking at the neurons and the neuroglia. We're also going to be looking at synapses and neurotransmitters. We're going to see kind of how all those different structures will work. We're going to look at the differences between an electrical and a chemical synapse. We're going to look at white matter and gray matter. And then we're going to take a look at how regeneration works, if it's able to work in the nervous system. Okay. So that's our goal for today. So just a quick little intro. The nervous system is going to be compared to almost like a supercomputer. And so a supercomputer, if you kind of think of what it does, it is going to be taking in lots of different inputs that we're going to be entering into the supercomputer. It's going to process all the information and then create an output, create some kind of action plan that needs to take place. The nervous system is going to work exactly the same way. Okay, it's going to be taking in lots of inputs from our internal and external environment. It's going to be able to process all the information, kind of facilitate it, integrate it, and then determine what it needs to do, and then initiate the action that will eventually be taking place. Okay, so it's going to be basically the big boss man for the whole body. It's going to be regulating everything that happens throughout the body. Okay. So when we look at the study of the nervous system, that is called the study of neurology. And so that's where you look at the function of the nervous system as well as the any disorders that are associated with the nervous system as well. Okay. Now, what's kind of interesting about the nervous system is that it only makes up about 3% of your total body weight. Okay. So it's a very small kind of a size in terms of its size. However, because it's constantly working, it needs a very large amount of blood. And so it actually takes about 20% of your total blood flow so they can get about 20% of your body's oxygen. Okay. And the reason for that is going to be because the brain and the spinal cord and all of the nerves are constantly working, even when you're asleep, when you're awake, even when you're just kind of in your empty box, nothing's happening. Okay, it's always going to be working regardless of what you're doing, and working a lot. And so it needs to have a very large amount of blood flow in order to help it do what it needs to do. Okay. And so for that reason, the nervous system is actually very, very sensitive to changes in oxygen levels. Okay. Okay, so when we take a look at the nervous system, there's going to be kind of 4 overall major components to it, and then we're going to see, or I guess 5 major components to it, and then 2 others kind of more sub-level kind of importance. The first one is going to be the brain, okay, and then the cranial nerves, which come off of the brain, okay, and their branches. Then we have the spinal cord and all the spinal nerves that come off of the spinal cord. Okay. We're going to see some sensory receptors that are going to be responsible for picking up on different stimuli from our internal and external environments. We're going to see some ganglia, and we'll look at what those are later. And then the enteric plexus, and we'll look at what that is in a second as well. Okay. But there's actually going to be 7 components here that are going to make up the nervous system and allow the nervous system to do what it needs to do. Okay, so we're going to kind of look at the nervous system kind of in its anatomical divisions here. And so the first branch of the nervous system as a whole is called the central nervous system. So I believe Tamim brought this up earlier. So this is the central nervous system. The central nervous system is going to be made up of the brain and the spinal cord. Okay, and between the 2 of those, there's over 85 billion neurons just within those particular regions. And that's not even taking into account all the nerves that come off of there. Okay. So about 85 billion neurons all making up the brain and the spinal cord. Okay. Now, these tissues are extremely, extremely sensitive. So the brain and the spinal cord don't like to have a whole lot of trauma. If there's a little bit of trauma to the brain or spinal cord, that causes a lot of significant damage, and the brain and spinal cord are not able to kind of bounce back from that as well. Okay? As well as other tissues in the body, like the heart, the lungs, things like that. Okay? So because they're so sensitive and because they're so easy to damage, they have to be protected extremely well. Okay? And so the way that they're going to be protected is going to be by the skull to protect the brain, and then the vertebrae to protect the spinal cord. Okay? So here we have our brain, and then our spinal cord, which should be running down our spine. That's going to be protected by the vertebrae. Okay? Now, the brain and the spinal cord, they're going to be connected to one another. Okay? And they're going to connect to each other through that big hole inside the occipital bone called the foramen magnum. Okay, so if you remember from looking at the bones of the skull, the occipital bone has that very large hole in it, the foramen magnum. Okay, that's going to be where the brain and the spinal cord are actually going to make direct contact with each other. Okay. So the central nervous system is really, really important because it's going to be regulating and kind of acting and determining what everything else in the body is going to be doing. And it's going to be able to process lots of sensory information. So any information coming into the brain, it's going to be able to process all of that. It's going to be responsible for initiating any motor movements. Okay? And then also it's going to be responsible for initiating different types of secretions from different glands as well. And then on top of that, it's going to be able to be the source where we're going to be finding all of our thoughts, all of our emotions, memories. It's also going to be the place where reflexes are going to initiate from. There's going to be pretty much everything that's going to be part of your executive functioning is going to be taking place within the brain and the spinal cord. So this is extremely, extremely important. Okay. All right, and the next component is the peripheral nervous system. So this is the second major division of the nervous system. So the peripheral nervous system is made up of everything outside of the brain and the spinal cord. Okay, so everything outside of the central nervous system is considered the peripheral nervous system, or the PNS. Okay. So the parts that make up the peripheral nervous system are going to be all 12 pairs of cranial nerves. Okay. So the cranial nerves are going to be the nerves that come off of the brain and branch up from the brain. Then we're going to have 31 pairs of spinal nerves, which will again branch off of the spinal cord. Okay. And so there's going to be one thing that we want to kind of keep track of here. And so when we talk about nerves and neurons, a lot of times we use those two terms interchangeably, which isn't technically correct. Okay, so a nerve is just a bundle of axons. Okay, so if we think of a neuron, that's going to be the individual cell within the nervous system. A nerve, so each, let me backtrack. So each neuron is going to have, dendrites, it's going to have a cell body, and it's going to have an axon. Okay. Now, a nerve is going to be made up of thousands of, or hundreds of thousands of different axons. So it actually takes up multiple neurons, okay, in order to form a single nerve. Okay. So a nerve is going to be a bundle of axons, as well as all the connective tissue and the blood vessels that help to, that hold everything in place there. Okay, that entire bundle of axons, connective tissue, and blood vessels is what we refer to as a nerve. Okay. Okay. So those are nerve components. We're also seeing some sensory receptors. So the sensory receptors are going to be kind of like on your skin that will be able to sense different amounts of pressure, pain, temperature, vibrations, things like that. So they're going to be able to take all of that information and then eventually send it to the brain. And so the whole goal of the peripheral nervous system is to provide a link between the central nervous system and the rest of the body. Okay? So be able to allow the brain and the spinal cord to communicate with your arms, your legs, your muscles, your lungs, your GI tract. Okay? Those are going to be all the different components that need to be able to communicate with the central nervous system. So they're going to rely on the peripheral nervous system to allow that communication to take place. Okay. Any questions so far on the central nervous system versus the peripheral nervous system?
No.
The peripheral, you said, is to allow the brain and spine to communicate. Is that what you said? Or
no. So it's to allow the brain and the spinal cord to communicate with everything else.
oh,
So it allows the brain and the spinal cord to then communicate with your muscles. Allow your brain and spinal cord to communicate with your like your lungs. Okay, allow them to communicate with your kidneys and your bladder and things like that.
got it.
So basically allowing it to communicate with the rest of the body.
Okay.
Yeah. Cool. Any other questions here? Okay, cool. Okay. So that's the anatomical organization of the nervous system. Okay. Now we're also going to see a functional organization of the nervous system. Okay. So the functional organization is going to be based off having some sensory function, some integrative function, and then some motor function. Okay. So we'll kind of take the example of let's say you've got some kids and you hear your kids screaming, right? So the first function that you're going to have when you hear your kids screaming, okay, that's obviously going to be the sensory component. That's going to be what you're actually able to sense. Okay? So the sensory receptors are receptors that are responsible for detecting different stimuli, be either in your internal environment or your external environment, and then sending that information to the brain and the spinal cord using the cranial or the spinal nerves. Okay? So for example, if you hear some kids screaming, okay, you're going to think, okay, there's some screaming kids. Okay? The nerves in your ears are going to be able to transmit that impulse to your brain. Okay? Because that's going to be the first component. Okay? That's going to be the sensory component. Okay? The second component is going to be the integrative function. Okay? So the integrative function is going to be responsible, or is going to be determined by the interneurons. Okay? So interneurons are these very short kind of axon having neurons here. Okay? And so these are responsible for integrating that sensory information, processing it, and then determining what to do. Okay? So if you hear some screaming kids, okay, that gets sent to your brain. Okay? The interneurons inside your brain and your spinal cord will then take the information that you just received and then process it. Okay, it's going to say, okay, are those my kids screaming? Okay, yes, those are my kids screaming. Are they screaming out of joy or are they screaming out of danger? Okay, they're screaming out of danger. Okay, so what is the appropriate response that I need to perform? I need to go run and check on my kids. Okay, so that's what the interneurons are going to be doing. They're going to be processing that information, okay, processing that sensory information that it just received from your sensory receptors and sensory neurons, okay, and then determine what type of response needs to happen next. Okay. Once the interneurons have determined what type of function needs to happen or what type of action needs to take place, okay, after the integrative process has occurred, it's going to then activate some motor neurons. Okay. And so motor neurons are going to be the neurons that are responsible for moving from the brain to the spinal, or brain to your muscles, okay, or your glands. So they're going to be carrying motor information from your brain and spinal cord to your muscles or to your glands in order to cause the actual action to take place. Okay. So we kind of take this whole example from beginning to end here. Okay. Our sensory input would be screaming kids. Okay. Screaming kids. That sensory information is going to run from our ears to our brain. Okay. Once we get to our brain, we're going to have our interneurons starting to integrate that information. So, okay, those are our screaming kids. They're screaming out of danger. I need to go run and help them. Okay. So then from there, the interneurons are then going to stimulate your motor neurons to go activate your legs, so your legs can help you get to your kids to then figure out what's going on, what the danger is, and then get them out of that danger. Okay. So the motor neurons are responsible for stimulating the muscle contractions or the glandular secretions that need to happen, okay, regardless of, depending on the situation. Okay. So that's kind of one example. Let's take another example here. So let's say we're going to use Kelly as an example. So let's say, so Kelly's not ratchet, but let's just say for the case of this example, Kelly's as ratchet as they come. Okay, so Kelly's our example here. Let's say Kelly, is walking down the street and someone pulls her hair. Okay, once Kelly gets her hair pulled, what's her first reaction going to be? What's the first thing that's going to happen? Which of these processes is going to be first? Yeah, go ahead.
So the sensory input, correct?
Right, she's going to have some sensory input.
Uh-huh.
Right. Her sensory receptor is going to say, hey, there's a lot of pressure here, a lot of tension. And then that information, that tension information, that pressure information, is then going to get sent to the brain and spinal cord, to the interneurons there, exactly. Okay. So then at that point, the interneurons can then process that information, okay? What just happened? Where did the tension come from? Came from my hair, okay? What happened? Someone pulled my hair. What am I going to do? Okay. So what's her reaction going to be here?
Motor neurons going to activate. It's going to activate the muscles to do whatever, to punch somebody, push, or
run away from it.
Right. Yeah. So let's assume Kelly's as ratchet as they come, right? So her interneurons going to say, hey, we need to start fighting. We need to start swinging. Okay. That's going to be the appropriate response that her interneurons might come up with here in this particular scenario. Okay. I know it's kind of out there, but just bear with me. Okay. So those interneurons are going to say, hey, we need to start swinging. So those interneurons are going to communicate with the motor neurons, okay, in her arms to start swinging. Okay. And then all of a sudden, now we're in a fight, game over. Okay. For the other chick, not for Kelly, because Kelly's a baller. Okay. Does that kind of make sense here in terms of the sensory, integrative, and then motor functions here?
Yes.
Yeah.
Yes.
All right, cool. Beautiful, beautiful. Okay, so those are the functional aspects of the neurons here. Okay. Now, when we're looking at the peripheral nervous system, so actually, let me kind of backtrack here. So this is all kind of a combination of what's happening in the central nervous system and in the peripheral nervous system. Okay. Now, when we're looking at the peripheral nervous system itself, the peripheral nervous system can itself be divided into a few different areas as well, a few different sub areas. Okay. The first of those sub areas is called the somatic nervous system. Okay. So the somatic nervous system is going to have its own sensory and motor neurons present here. Okay. So the somatic sensory neurons, okay, those are responsible for sending sensory information from the skin, from the muscles, and from your special senses. Okay. So when you think somatic, think kind of body, think kind of more like action stuff. Okay. Your skin, muscles, senses like your vision, touch, auditory impulses, things like that. Okay.
Doesn't soma
it's gonna be
stand for
sending-
skin?
Yeah. Yeah, so it is going to be helping with the skin as well.
Correct?
Yes.
Okay.
Yeah, so the somatic sensory neurons are going to be sending information from your external environment, okay, up to the central nervous system, okay, up to the brain and your spinal cord here. So that's going to be the input component. Okay. From there, once the brain processes all that information, it's then going to produce a motor output, okay, using some somatic motor neurons. Okay. So the somatic motor neurons are going to come from the brain and spinal cord, okay, and they're going to stimulate skeletal muscles only, okay, in order to provide some kind of motor movement. Okay. So an example here is we got a guy who put his finger on a flame. Okay. So the sensory input is going to be, hey, this is really hot. Okay. So it's going to send that information, that heat information, to the brain and spinal cord. Okay. From there, the brain and spinal cord is going to say, hey, this is too hot. If we keep our hand here, we're going to end up burning ourselves. We need to move our hand away. Okay. And so the brain and spinal cord are then going to stimulate the somatic motor neurons to his muscles in his arms to help him pull his hand off of that flame. Okay. And so these are voluntary actions. So the somatic nervous system is responsible for voluntary movements, okay, voluntary actions here.
So there are no interneurons involved in this process, correct?
In some cases, there can be. It depends on the situation. In this case, because we're looking at a reflex, there wouldn't be any interneurons, and we'll look at that on Wednesday. But in other cases, like for example, if you see a pencil on the floor and you want to pick it up, okay, then in that case, you would have interneurons being activated there.
I see.
Yeah. Yeah. So, and again, we'll talk a little bit more in detail about that on Wednesday too. Okay. That's the somatic nervous system. Those are things that we can voluntarily control. The second component of the peripheral nervous system is called the autonomic nervous system. And we'll look at this in a lot more detail in 2 weeks in chapter 19. So the autonomic nervous system also has sensory and motor components to it, but it's going to be specifically responsible for controlling your visceral organs. Okay, so controlling all the organs in your thoracic cavity as well as in your abdominal cavity. Okay, so it's going to be responsible for your lungs, it's going to be responsible for your heart, your GI tract, your bladder, the reproductive tract. Okay, all of those different organs in those cavities, okay, are all going to be regulated by the autonomic nervous system. Okay, so it's going to be referred to as autonomic because these are things that are going to be happening automatically, things that you don't have to think about. These are going to be things that are involuntary. Okay, so the sensory neurons that are inside the autonomic nervous system are sometimes called autonomic sensory neurons. Sometimes they're called visceral sensory neurons. But their job is to take a lot of sensory information from those organs and then deliver them to the brain. Okay. So for example, let's take the lungs, for example. Let's say our lungs have expanded. Okay. There's going to be specific neurons inside the lungs that are going to help to tell the brain, hey, our lungs are expanded here. Okay, now it's time to breathe out so we can bring our lungs back to their original shape. Okay. So they're going to be able to take in lots of sensory information from various different organs, okay, provide that information to the brain. Once the brain receives that information, it will then activate or not activate that that particular organ using autonomic motor neurons. Okay. So the autonomic motor neurons are going to be where the central nervous system will physically control the smooth muscle, cardiac muscle, or glands of some organ inside your viscera. Okay. And so that's going to cause those muscles to either contract or relax, or cause the glands to secrete or not secrete. So again, these are going to be involuntarily controlled parts of the nervous system. Okay. So when we look at the autonomic nervous system, it's going to be divided up into two branches. Okay? The first branch is the sympathetic division, or the sympathetic branch. The sympathetic branch is our fight or flight responses. Okay? It's responsible for the fight or flight responses. Okay? So, for example, if if you go camping and you see a bear, okay, your first response is going to be, "Hey, I need to get away from this bear." So that's going to be where the sympathetic nervous system, or the sympathetic division of the autonomic nervous system, kicks in. Okay? So it's going to be responsible for helping you to get out of danger. Okay? It's going to help you with emergency situations or in exercise situations as well. Okay? The goal of it is to increase your heart rate and then be able to help you escape from that particular danger or that threat, whatever it may be. Okay? The flip side to that is the parasympathetic division. Okay? And so the parasympathetic division is responsible for your rest and digest responses. Okay? So when you've just eaten, or when you're about to eat, or when you're just taking a nap, you're sleeping, whatever you may be doing, that is going to be responsible by, or that's going to be taken care of by the parasympathetic division. Okay? So the goal there is to reduce your heart rate to allow you to just kind of relax and then allow your GI tract to be active. Okay? Now, most effectors, and so effectors are anything that's going to be stimulated by the nervous system. So that can be muscles, can be glands, organs, whatever it may be. Most effectors are going to have some kind of stimulation from either the sympathetic or the parasympathetic. And in many cases, they can be stimulated by both, okay? Both the sympathetic and parasympathetic division. However, only one of the two can be active at any point in time, okay? You can never have your sympathetic division and your parasympathetic division both be active, because they're going to be countering each other. Okay? So each organ is going to be stimulated by the sympathetic division at one point in time, or it'll be stimulated by the parasympathetic division, okay? But never both at the same time. It's either one or the other. Okay. Does that make sense?
Yes.
Yeah. So there's okay. So there's two parts: the autonomic nervous system.
Yeah.
Autonomic. Okay. There's
Mhm.
two parts of that,
Yeah.
right? So it's a sympathetic division
Right.
and the parasympathetic. Okay.
Mhm.
Exactly.
What's a visceral organ? Like, what does that mean?
So a visceral organ is just any organ inside your thoracic or your abdominal cavity. Okay?
Oh,
So your lungs, heart, stomach, kidneys, pancreas, anything that's inside your thoracic or abdominal cavity is considered a visceral organ.
Okay.
Non-visceral organs would be like your muscles, your eyes, your brain, things like that.
Oh.
So viscera is just inside your chest and your abdomen.
Okay, got it.
Yeah. Mm-hmm. Okay. All right. Then the last component of the peripheral nervous system is the enteric nervous system. Okay. So the enteric nervous system is specifically for the GI tract. Okay. So we call this division kind of like the brain of the gut. Okay. Because it's responsible for causing all the different things that need to happen inside of your GI tract. Okay. So we have 100 million neurons specifically found inside the gastrointestinal tract, and just like the other ones, we're going to have some sensory and motor neurons present as well. Okay. So the sensory neurons here are going to be responsible for being able to pick up on chemical changes inside the different organs, and then also being able to determine the amount of stretch that's happening inside the walls of those organs. So for example, let's say it's Thanksgiving and we just eat to our heart's content. Okay, we're going to have lots of food inside of our stomach. Okay? So the sensory neurons inside of the stomach are going to be being able to pick up on the amount of stomach acid that's present. That's going to be the chemical components. And then it's also going to be able to determine the amount of stretch that's happening inside the stomach. It'll be able to tell you that the stomach is stretched as far as it can possibly stretch because you filled it with all the food it can hold. Okay. So those are things that are going to be picked up by these sensory neurons. Okay. The motor neurons here are going to be responsible for causing the GI muscles, or the GI muscles to contract. Okay. So the smooth muscles of the GI tract are going to contract, and that's going to be helping to move the food through the GI tract, and then also help to produce different secretions of the GI tract as well. Okay. Okay. And so again, as you can probably imagine, these would be involuntary secretions because we cannot physically control what our enteric nervous system is doing. Okay. That is the enteric nervous system. Okay. So this is just a little chart here that kind of shows kind of the connections between the central and peripheral nervous systems in the different kind of components here. Okay, so if we look at the somatic nervous system of the peripheral nervous system, we're going to say our somatic sensory receptors are going to send information to the central nervous system. The CNS is then going to process all of that and then determine what types of motor actions need to happen. Okay. So it's going to kind of go back to the somatic nervous system using the somatic motor neurons, okay, to stimulate your skeletal muscles here. Okay. In the case of your organs, and with the sympathetic and parasympathetic divisions, we can have our visceral sensory receptors, again, send information to the CNS. Okay. From there, the CNS will process that information and then refer back to it using the autonomic motor neurons in order to stimulate the smooth muscle, cardiac muscle, or glands. Or it can actually impact the enteric nervous system, okay, and trigger motor contractions happening here in terms of smooth muscle, glands, and then the secretions from the GI tract as well. This is another kind of diagram to kind of help kind of with the breakdown of everything. So you have your total nervous system, which is going to be divided up into the central nervous system, which is your brain and spinal cord, your peripheral nervous system, which is everything else. From there, the peripheral nervous system can then be divided up into the somatic or the autonomic nervous systems. Okay. And then the autonomic nervous system gets broken down into the sympathetic or parasympathetic. Okay. And so that's how the entire nervous system is organized. So a question here. So which of the following are structures of the nervous system? So we got the spinal cord, the ganglia, sensory receptors, the enteric plexuses, or all of the above?
I think it's all of the above choices,
All
right?
the above for the
All the
nervous.
above.
Yes, all of the above
above.
is absolutely correct. Perfect. So these are all components of the nervous system. Okay, so next one. So which type of neuron functions to integrate or process sensory information? Is the efferent neuron, sensory neurons, interneurons, motor neurons, or afferent neurons?
D.
Interneuron?
Which type of-
Yes. Yeah, absolutely. So interneurons. So interneurons are responsible for integrating all that information. Okay. So one way they kind of remember that is integrate, and interneuron can kind of begin with the same few letters. So they're going to be, that's going to be the role of the interneurons there. Perfect. All right. Next one. So the peripheral nervous system, the PNS, is divided into which three functional components? So we got A, the peripheral autonomic and enteric nervous systems. B, the sympathetic, autonomic, and enteric nervous systems. C, the parasympathetic, sympathetic, and spinal cord. Or D, the somatic, autonomic, and enteric nervous systems.
E.
D.
So,
I think it's D. Yeah, D.
Yes, so D is correct, right. So the somatic, autonomic, and enteric nervous systems are the three overall functional components of the PNS. Perfect. All right, cool. Any questions so far?
No.
No? Okay. All righty. Okay, so with that, we're going to take a look at the nervous tissue. Okay? So nervous tissue, like we talked about before, is going to be constantly active. Okay? And so because it's constantly active, there has to be lots of blood vessels available to the brain and the spinal cord and all the rest of the nervous tissue as well. Okay? So this is going to be a very highly vascular tissue because it needs a lot of oxygen to make sure that it's able to do the very extensive functions it needs to be able to perform. Okay? So highly vascular tissue. Now, in addition to that, we're also going to find two different types of cells inside our nervous tissue. Okay? The first type is going to be the neurons, okay? And then we're also going to have some neuroglia. Okay? So neurons are also called nerve cells. Okay? Neurons are responsible for producing all those action potentials, all those electrical impulses. Okay? So these are very highly specialized cells. They have to be able to produce action potentials constantly throughout the day in order to make sure that the body is working the way it needs to work, and to make sure that everything, all the functions inside of the brain and inside of the spinal cord are operating the way they need to work as well. Okay? So they're highly specialized cells because they're going to be producing lots of different electrical impulses. Okay? Now, these neurons are going to be allowing the brain and the spinal cord to connect not just with themselves, okay, but also connect to the rest of the body as a whole. And so it's going to be able to provide kind of the electrical circuitry of our kind of supercomputer here. Okay, it's going to be providing the circuitry that helps to connect, the brain to the rest of the body, to connect the spinal cord and the rest of the body, and then allow the brain and the spinal cord to communicate within themselves and then with each other as well. Okay. So lots of different types of connections are going to be happening based off of the actions of the neurons. Okay. Now these neurons are going to be, they're going to be kind of really, really important here because they're going to be doing a lot of different types of functions. The problem here is that these guys cannot undergo mitosis, so they cannot divide. And that's often why, if you ever ride your bike or if you have a motorcycle, it's required and almost kind of like really encouraged for you to always wear a helmet. Okay? The reason for that is because the neurons inside of the brain and the spinal cord are very, very, very sensitive. Okay? And if they get damaged, it doesn't take a lot to damage them. If they get damaged, there's no way for them to regenerate, okay, because they cannot undergo mitosis anymore. Okay? So the brain cells that you have is all you're ever going to have. So you got to take care of it to the best of your ability as much as you possibly can. So they cannot undergo mitosis. They cannot undergo division. Okay? So you cannot replace these guys if they get damaged. Okay, so those are the neurons. Okay? The second type of cell here we're going to find are the neuroglia. And so with as many neurons as we have an even more neuroglia. And the neuroglia are these much smaller cells, but they vastly outnumber the amount of neurons that we have. Okay? And so contrary to the neurons, their job is not to produce any action potentials. Okay, they're not producing any electrical impulses here. The only thing that the neuroglia do is that they're going to support, nourish, and then protect the neurons. Okay? They're going to support them and make sure that all these neurons are able to do all the numerous types of functions that they need to be able to perform. Okay? So they allow the neurons to be able to do what they do. Okay? The flip side is that these guys do have the ability to divide. Okay? So neurons themselves cannot undergo division, okay? But the neuroglia can divide extensively throughout an individual's lifespan. So those are those guys. What we can actually see here, so here we have a slide of a neuron. Okay, you can see this, all the different components of our neuron here. All of these little dots kind of surrounding the neuron, okay, these are all individual different types of neuroglia. Okay? So these are going to be the cells that actually support all of those neurons. Okay? They look very small and almost you would probably even miss them if you didn't know you were looking for them, but they have a very, very important role inside the nervous system. Okay, so we're going to start by taking a look at neurons. Okay? And so neurons, like we talked about before, have a lot of electrical excitability, okay? Which means that they're going to be able to respond to different types of stimuli, okay, by producing an electrical impulse. Okay? That electrical impulse is what we refer to as a nerve impulse or an action potential. Okay? So an action potential is going to be an electrical signal that's going to travel along the plasma membrane of a neuron. And so if we remember from the neuromuscular junction, okay, I'm gonna kind of draw out a little bit of a neuron here. Okay. If we remember here, all of these cells are gonna be mostly negatively charged. Okay. For an action potential to take place, we need to switch this from being negatively charged to positively charged. So an action potential is essentially a change in voltage, okay, from a negative to a positive charge inside your neuron. Okay. Now the way this happens, and I'll go over it today so we have an idea of exactly what's happening, but you don't really need to worry about it too much until you take physiology. But essentially what happens here in order to create this positive charge is we're going to have an influx of these sodium ions. So sodium ions are positively charged, and when they enter an axon or enter a neuron, okay, through various types of channels, okay, that's going to cause the voltage to become positive, okay, switches the voltage, and now you're able to generate an action potential. Okay. Does that kind of make sense so far?
Yes.
Kind of follow me?
Yes.
Yeah.
Yeah. So sodium ions, and then to a lesser degree, potassium ions as well. But sodium ions are really going to be the main component here that's going to allow these action potentials to take place, okay, by increasing the voltage from negative to positive. Okay. So keep that in mind. We'll kind of play with that a little bit more as we go forward here. Okay. Okay. So that's going to be how we generate an action potential. Now, when we generate that action potential, they're going to be moving extremely fast, and they can actually move up to a speed of about 280 miles per hour. Okay. So they move incredibly fast through the neurons. Okay. And we'll take a look at why exactly it's able to move so fast in a second here too. Okay. Okay, here. Okay, so we're going to take a look at the parts of a neuron. And so the first part of a neuron is going to be the cell body, or also called the soma. Okay. So the cell body is going to be a part of the neuron that's going to contain the nucleus. It's going to have the cytoplasm, and then the rest of the organelles that are going to be present. Mitochondria, Golgi apparatus, rough ER, smooth ER, all those other organelles are going to be found inside of the soma, or the cell body. Okay. Also within the cell body, we're going to have a particular structure called the Nissl bodies. And so the Nissl bodies are these very large clusters of rough endoplasmic reticulum. Okay. And so because we have very large clusters of this rough ER, we're going to be able to produce very high levels of protein synthesis. Okay. That's going to be really important because that protein synthesis is going to be important for allowing us to be able to maintain and repair small damage within the neurons. Okay. It's going to help with allowing action potentials to transmit effectively through the neuron. Okay. And then it's also going to be responsible for helping to allow different stimuli to be received by that neuron as well. So Nissl bodies are just very large clusters of rough ER to produce large amounts of protein. Okay, so that's that. Okay, now coming off of the cell body, we're going to find a number of nerve fibers, which are these long processes that come off of this cell body. Okay. The first type of nerve fiber we're going to see are the dendrites. So dendrites tend to be relatively short processes, but they're very highly branched. And so these act as the receiving components, or the receiving ends of the neuron. Those are going to be the sites that are going to receive all of those sensory or receive different types of input, okay, from the neuron. Okay. And so because they're very extensively branched, that is going to increase the surface area dramatically of our dendrites and of our neuron here, okay, of the sensory side, okay, or the receiving side, I should say. Okay. With a higher surface, with a higher surface area, this allows a neuron to be able to communicate with as many other neurons as possible, okay, and create lots of other synapses and lots of other points of communication with other neurons. Okay. The higher the surface area, the more neurons you can communicate with, okay, which is going to be really important for a specific different types of neurons here. Okay. Another thing that's going to be important here is that on the plasma membrane of all of these dendrites, we are going to have all the receptors for our neurotransmitters. Okay. So this is what we see here. Okay. So here we have our synapse. Okay. Here we have our neurotransmitters. Our neurotransmitters up here, and our neurotransmitter receptors. Because this is the receiving side, this is going to be the side that's going to be receiving the signal from a previous neuron. Okay. Those receptors are usually going to be on the dendrites of the receiving neuron. Okay. And we'll look at that in a little more detail as we go forward here. Okay. So dendrites are going to be the input side of your neuron. And that's going to be the side that receives different signals. Okay. On the other side of the neuron, we'll typically have a long singular nerve fiber, and that is called the axon. And so the axon is a very long, typically very thin and kind of cylindrical structure. This is going to be where we're going to generate action potentials from a particular neuron here. And so that action potential is going to generate at the axon. It's going to move down the length of the axon and eventually reach another neuron, a muscle fiber, or another gland. And so the axon is going to begin at a region called the axon hillock. So when we look at the different parts of an axon, the first structure that we're going to see is called the trigger zone, which is in between the axon hillock and the actual axon itself. That particular point is going to be where the action potential actually generates from. Okay? So it would generate from a spot right about here. Okay? Once we produce that action potential, it will then move down the length of this axon until it reaches the end of it. Okay? A couple other regions, we're going to have the axoplasm, which is just the cytoplasm inside the axon, the axolemma, which is just the plasma membrane of the axon. Don't worry too much about those. But then at the end of it, we'll have the axon terminal. And so the axon terminal are these processes at the end of the axon, okay? And that is going to be where we start to form some communication sites between this neuron and the next neuron in the chain. Okay. All right. Any questions so far on the structure of a neuron here?
No, not right now. No questions.
Okay. Okay.
Good.
So the axon is going to be the output region of the neuron. The dendrites are going to be the input region of the neuron. Okay. So this is just a larger picture, kind of more detailed picture to show you kind of what this whole thing would look like in more detail. Here again, you can see all the dendrites coming off of our cell body. Here's the cell body itself. We see the axon hillock right about here. And then we have our axon coming down this region here. Okay. And then at the end, we eventually will reach our axon terminals. Cool. Okay. So now that we've kind of gone through the different parts of an individual neuron, okay, we're going to look at the site where they actually communicate with one another, where you have two neurons communicating with one another. That region is called the synapse. Okay? So a synapse is a region where either two neurons are going to interact with one another, or you have a neuron interacting with some effector, okay? Either a muscle or a gland or whatever it may be. Okay? That's going to be the site where that communication will take place. Okay? So for a synapse, there's going to be three major components. Okay? The first component is going to be the synaptic cleft, which is just the space in between the two neurons here. The second component is the presynaptic neuron. So the presynaptic neuron is the neuron that comes before the synapse, which is going to be the one that takes a nerve impulse towards the synapse itself. Okay. Then we have the postsynaptic cell. So the postsynaptic cell can either be a neuron, it can be a muscle, or it can be a effector. In our case, for now, we're just going to assume that our postsynaptic cell is a neuron. So we'll just call this a postsynaptic neuron here. Okay. So the postsynaptic neuron is going to be the neuron that comes after the synapse. Okay. So the signal will come from the presynaptic neuron, okay, run through the synapse, and then eventually reach the postsynaptic neuron. Okay. And then from there, that action potential will then move from the postsynaptic neuron down towards the rest of that particular neuron there, the cell body, and so on and so forth. Okay. So presynaptic neuron sends a signal, the postsynaptic neuron receives that signal. Okay. Now at the end of each axon, you're going to have this swelling here, okay, this part that kind of bulges out, and this is what we refer to as a synaptic end bulb. Okay. The synaptic end bulb is going to contain a series of vesicles, okay? And these vesicles will contain neurotransmitters, okay? which we see over here. Okay. These neurotransmitters are going to get released, and once they get released, they're going to either excite the next neuron to either stimulate an action potential in the postsynaptic neuron, or they're going to inhibit that signal from taking place, okay? And prevent that signal from occurring. Okay. Either onto that neuron or a muscle fiber or gland, whatever it may be. So we've seen a neurotransmitter already when we were looking at muscle contractions, and so this one that we saw was acetylcholine. Okay, but there are a number of other neurotransmitters, which again we'll take a look at in a little bit later. Okay. Okay. So we have three overall types of synapses here. Okay. The first type of synapse is called an axodendritic synapse. So an axodendritic synapse is a synapse between a presynaptic axon and then a postsynaptic dendrite. Okay. So in this case, whenever you have your neurotransmitters getting released, the receptors are going to be found on the dendrites here. Okay. So neurotransmitter receptors will be found on the dendrites here. Okay. So this is an axodendritic synapse. Okay. These make up the vast majority of synapses in the nervous system. We can also have axosomatic synapses. Okay? And so axosomatic synapses are synapses between a presynaptic axon and then the soma of a postsynaptic cell, or the cell body of a postsynaptic cell. So that's kind of where I get this name from, axosomatic. Okay, so in this case, the neurotransmitter receptors will be found on the cell body itself. Okay. And then the last type that we can have are called axo-axonic synapses. And so these are where we have a presynaptic axon synapsing with a postsynaptic axon. Okay. So those are the three overall types of synapses that we can have in regards to releasing neurotransmitters. Now, there's kind of two overall types of synapses like we just talked about. Okay, the one that we just talked about was called the chemical synapse. I'll give a little more detail on that in a second. Chemical synapses involve neurotransmitters. We also can have electrical synapses, which are a little bit more rare in the nervous system, but they're also found throughout the rest of the body. And so these will usually require gap junctions. Okay? So in an electrical synapse, you usually have an ion running from one cell through a gap junction into the next cell. Okay, so they're just running straight through from one cell into the next using those gap junctions. So these are typically going to be common in cardiac muscle, like we saw with the intercalated discs. They're going to be found in smooth muscle to some degree, and then in embryos. And because you just have ions moving from one cell directly into the next one, the communication between those cells is very, very quick, extremely rapid. And so this allows lots of uniform, very coordinated movements in a very short period of time. The ones we'll be focusing on the majority of the time for our nervous system here is going to be the chemical synapses. And so these ones will involve neurotransmitters. Okay? And so in this case, because we need neurotransmitters to be released and then end up binding onto the receptors, it takes a little bit longer for these for this communication to occur. Okay. But the key here is that you have to generate action potentials in one cell and then generate a whole new action potential in the next one. Okay. Action potentials cannot just jump from one cell to the next one because there's a physical space in between the two neurons. Okay. You have that synaptic cleft between the two. Okay. Action potentials cannot jump across the synapse because it's almost like trying to jump the Grand Canyon. It's too big. Okay. And so in order to generate that action potential, you have to use these neurotransmitters. Okay. These neurotransmitters have to allow the postsynaptic cell to generate its own brand new action potential. Okay. And so we'll look at that in a little more detail in a second here. Okay, cool. Okay, so this is what a typical synapse would look like. And kind of just looking right at this picture here, we have seen this picture before, and it should look pretty familiar. Okay, where have we seen this picture before?
That's the incredible one.
No, we've seen it in previous chapters.
Yeah.
Yeah,
Was
in
it
previous
the
chapters.
muscle?
It was like a chapter ago or so.
Right.
Was it the muscles?
Yes,
So,
so exactly right.
muscle.
So when we were looking at muscle contractions,
Yeah.
right?
There we go.
So
Yep.
we were looking at the neuromuscular junction where neurons are going to communicate with a muscle fiber in order to trigger a muscle contraction. Okay. Now, really cool thing here is that synapses between one neuron and another neuron work exactly the same way with a slight little caveat that's a little bit different at the end of it. Okay. So if you didn't learn how the neuromuscular junction worked on the previous test, okay, this is your chance to redeem yourself. Okay, because this is going to be where it's going to be the exact same process. Okay. Exact same process.
Awesome.
Again, with a little bit of a caveat at the end of this.
That's the best.
And one other thing is you will see this again on a free response question on the next exam. So again, if you didn't get it the first time, this is your chance to get it again this time.
Oh, there
Yes,
you go.
exactly.
There you go.
So we'll kind of run through this whole situation one more time. So in order for an action potential to occur, what we need is a communication at a synapse to occur, I should say. Okay. What we're going to have is an action potential coming down the length of our axon here, right? Our presynaptic axon. Okay.
Mm-hmm.
Once we have this action potential occur, this is going to shift the charge from being negative to positive inside of our neuron. Okay. That shift in the voltage will cause our voltage-gated calcium channels to open. Okay. When our voltage-gated calcium channels open, calcium rushes in, and then what does that calcium do? What does it trigger?
Sodium ions?
The vesicles,
Yeah.
the exocytes of vesicles, correct?
Right. It's going to cause these vesicles to undergo exocytosis. And we're
Okay.
going to release our vesicles into the synaptic, sorry, we're going to release our neurotransmitters,
Synaptic cleft.
which are contained inside those vesicles, into
Yeah.
the synaptic cleft. Right.
Yep.
Okay. So once we release these neurotransmitters, okay, these neurotransmitters now inside the synaptic cleft are now going to do what?
They're gonna open.
They trigger the opening of
Yeah.
ion channels.
Isn't it calcium
It's sodium
ion?
ions.
Oh, sodium.
No,
I
no.
thought it was calcium.
Perfect, on the same page here. So they're going to bind
Okay.
onto these receptors,
Mm-hmm.
right? So these neurotransmitters are going to bind onto their specific receptors. Once they bind, those receptors will then open up and start acting as ion channels.
Ion
Okay.
channels.
There we go.
So they're going to act as either sodium channels, potassium channels, or chloride channels. This was the caveat I was talking about. Okay. So
Yeah.
depending on the ion that comes through, okay, that can either stimulate that particular neuron to either produce its own action potential, okay, trigger another action potential here and excite that neuron, or it can inhibit that nerve impulse, okay, and stop an action potential from occurring here. Okay. So I'm gonna kind of draw out how this whole process would work in these three kind of scenarios here. Okay. So let's say we're looking at sodium. I'll kind of draw out our, say this is our DEDs
Ion
right
beta
here.
channel.
Ooh, let me try that again. Okay. Ooh, okay, let's just assume that's our side neuron. Okay. Here we have a channel. Okay, and this is going to be specifically for sodium. Okay, and so when sodium floats in, so sodium typically wants to flow into the cells. Okay, so when it flows into this particular postsynaptic cell here, okay, is our voltage going to increase or is it going to decrease?
It's gonna go from positive, well, from negative to positive. So it's gonna
Right, it's
increase.
going to go from negative to
Increase.
positive. Right, because this was initially negative, right? It was initially negative. We have a lot of sodium ions rushing in, so the voltage is going to switch to being more positive. So I'm going to kind of draw out a little bit of a chart here.
Okay.
So let's just say this is negative. It's going to rise up and then become more positive. Okay. So in order to trigger an action potential, there's a specific threshold that you have to reach, which is right about here. Okay. Again, don't worry too much about this. This will make more sense when you take physiology. But essentially, you have to pass the threshold in order to create an action potential. Okay. So you need your voltage to become more positive. So in this case, we would have an action potential occur. So I'll put AP, and then I'll put a check mark. Yeah. So we have action potential, all good here. Okay. Makes sense so far.
Sure.
Mm-hmm.
Yeah. All we did was increase the charge here from negative to positive. Now, this is where things get a little more complicated. I'm going to use chloride as our next example. Okay, so chloride, let's say our axon in question here, sorry, our ion in question is chloride.
Mm-hmm.
So chloride also wants to flow into cells. Okay, so chloride is negatively charged. So if chloride comes into our postsynaptic cell here, okay, what's going to happen to our voltage? Is our voltage going to increase or is it going to decrease?
Decrease.
Yeah.
Decrease, right. So it's going to decrease. And so we never come anywhere near the initial threshold we needed, right? And so would we get an action potential at this point?
No.
No.
Actually, no.
No.
No, exactly. So you would have no action potential. And so this would be a way that you could kind of regulate
And
when you
inhibit,
can start
right?
a synapse or when you can start a signal or stop a signal. Okay. So we would have no action potential here. Okay. Does that make sense?
Yes.
Yes.
Yeah,
Mhm.
okay. Next one is potassium. I'll use a different color for potassium here. So potassium, when these ion channels open, potassium typically wants to leave a cell. Okay? Sometimes it can enter, but usually it's going to want to leave a cell. Okay? So if potassium leaves the cell, is our voltage going to become more negative or more positive?
More negative, right?
More negative, exactly.
Yeah.
So it's going to become more negative. Okay. And so again, that would be another way that you can stop a signal in your kind of neuron chain here. Does that make sense?
Yes.
Yes.
So depending on the ion that ends up flowing into the cell or flowing out of the cell, okay, that is going to help you regulate whether you continue a particular signal from one neuron to the next, or where you stop that signal from one neuron to the next. Okay. Okay. Any questions here?
So just to clarify, you need to go over the threshold in order to have an action potential, right?
Right.
Okay.
Yeah. You need to go over that threshold, and the only way that you can do that is to go from being negative to more positive.
Positive. Okay.
Yeah. Okay. Again, that's probably a little bit more detail than you need to know, but when you take physiology next semester or whenever it is, you'll have this kind of background information in the back of your mind, so you kind of have the idea behind it.
Okay. Thank you.
Okay. Cool. All right. So that is how our synapses are going to be working here. Okay. So this is just a larger picture. So again, to kind of run through it one more time, we have our action potential coming down our axon. Okay. That action potential is going to open up our voltage-gated calcium channels. Okay. Calcium is going to rush in and trigger the exocytosis of all of these synaptic vesicles containing our neurotransmitter. The neurotransmitter gets released into the synaptic cleft. It eventually will bind onto those neurotransmitter receptors. Okay. Once they bind onto the receptors will then act as ion channels and open up, okay, to allow that particular ion to flow into the postsynaptic neuron here, okay, whether that's sodium, chloride, potassium, whatever it may be.
Yeah.
All right, cool. All right, now we've talked about how we're going to be releasing our neurotransmitters. We also need to stop and look at how we can remove our neurotransmitters. Okay? So if we're in a situation where we want to stop a signal, okay, from taking place, we can remove a neurotransmitter in one of three ways. Okay? The first way is that we can just have our neurotransmitters just diffuse away from the receptors. Okay? So if they just move away, okay, they're no longer bound to the receptors, and then as a result, you're no longer going to have the effect. Okay? So the only way a neurotransmitter can trigger an effect is if it's bound to the receptors. Okay? If the neurotransmitters just diffuse away from the receptors, okay, then you end up stopping the effect, and then you're able to regulate your signal that way. Okay? That's option one. Okay? Option two is you can destroy the neurotransmitter using some enzymes. Okay? And so we saw this a few weeks ago when we were looking at acetylcholinesterase. Okay? So in order for a muscle contraction, for example, to occur, we need acetylcholine to bind onto these acetylcholine receptors. Okay? We want our muscle to relax. We're going to use this enzyme called acetylcholinesterase, or ACHE, okay? And it'll break down acetylcholine into an acetyl and then a choline group. Okay? And so once they split up, okay, once we destroy that particular neurotransmitter, it's no longer going to be able to bind to the receptors. Okay? You have no neurotransmitters binding to the receptors. You end up stopping your effect. Okay? So that's the second way of being able to remove your neurotransmitters here. The third way that you can remove your neurotransmitters is by a process called reuptake. Okay? And so reuptake is a process where you release your neurotransmitters, okay, they bind to the receptors, then instead of destroying them or removing them from the site, you basically pump them back into the presynaptic neuron and then repackage them into vesicles. So you're almost like recycling your neurotransmitters here. Okay? So typically they'll be reuptaken back into the, I think that's a word, reuptaken back into the presynaptic neuron. Okay? Or in some cases, they can be reuptaken back into the neuroglia. Okay? So those are the three ways. Okay? You can either cause those neurotransmitters to just diffuse away. You can destroy the enzymes destroy the neurotransmitters using an enzyme here, or you can pump it back into the original cell, into the original neuron using reuptake. Okay? In either case, or regardless of the case you're using, you're removing your neurotransmitters away from these receptors, and so you end up stopping the effect. Okay. Okay. Any questions on these three processes here?
No, I'm
No.
good.
No? Okay. Okay. So a couple ways where this can actually become clinically important here. So there are some toxins that are capable of stopping or blocking nerve impulses. Okay, one of those toxins is called tetrodotoxin, or TTX. Okay, and so tetrodotoxin is a toxin that's found inside jellyfish, or sorry, not jellyfish, pufferfish. Okay, and so I think it's in Japan. I believe it's Japan. Eating pufferfish is kind of like a delicacy. Okay? The problem with eating pufferfish is that you have to have a chef that's very skilled in cooking pufferfish, because if you don't cook it right, okay, the tetrodotoxin remains active. Okay, if that tetrodotoxin remains active in that food, okay, we end up eating that tetrodotoxin, that TTX, and so what it'll do is it'll actually block the sodium receptors, or sodium channels, that would produce your action potentials. Okay, so the sodium ions that help you to increase your voltage here, and these channels are going to get blocked, okay, by the tetrodotoxin. Okay, once those neurotransmitter receptors once those ion channels get blocked, okay, you stop your action potential. You're no longer able to release your neurotransmitters. Okay, and so that stops you from being able to have communication from one neuron to the next, or from one neuron to a muscle cell, and things like that. Okay? And so this is where things can get a little bit dangerous if you don't cook pufferfish right in that particular part of the world. Kind of interestingly, anesthetics also work exactly the same way. Okay? So when it comes to blocking pain impulses, okay, what dentists or other doctors will do is they'll give an anesthetic. Okay? The anesthetic will go specifically into a nerve, okay, block the ability of that particular neuron, okay, from communicating with the central nervous system. Okay? So if the brain and the spinal cord never gets a chance to process any pain stimuli, they're not going to sense that there's any pain to begin with. And so you end up having that numbing feeling. Okay? So that's kind of how anesthetics will typically work, okay, all by blocking these action potentials in various parts of the body. Any questions here? No? Okay. All right, so a question here. So in the figure shown, what is the indicated structure? Okay, so what are we looking at here? Is this our dendritic gemule? Is this our Nissl body, axon terminal, axon hillock, or the node of Ranvier?
Hillock, wasn't
Yes, that's the axon hillock, right?
Yes.
So this is the axon hillock. This is where we would be generating our action potentials. Perfect. Okay. Okay, so now taking a look at neurotransmitters, there's about 100 different substances that we know are, or that we suspect to be neurotransmitters. We looked at one of them, which was acetylcholine a few weeks ago. Oh yeah, go ahead.
Yeah. So when you're going through maybe the dental process of removing the wisdom teeth, sometimes they tell you that you might lose the sensitivity of the jaws, your mouth.
Yeah.
Does it have to do with what we are talking about?
Yes, absolutely. So, so Sharon's question, I believe that's Sharon. So her question was, why is it that, like, for example, if you get a local anesthetic when you get your wisdom teeth pulled, that the rest of your jaw kind of becomes more numb as well? Okay. The reason is because those synapses are still going to be running through the other neurons within your jaw as well. So even though you stop the impulse at one neuron, okay, stopping it at one impulse, or stopping that action potential at one neuron, okay, will stop it from communicating with the next one, and the next one, and the next one, and the next one. Okay, and so your brain never gets any of that particular pain, okay, or never gets any of those pain signals, okay, because you've numbed not just one impulse, but you've stopped it from communicating with other neurons in the chain as well. Yeah, good question. Very good question. Okay, so with neurotransmitters, we've got multiple different types of neurotransmitters. We've seen acetylcholine already. There's a bunch of others. So some other ones that you might be comfortable, or that you might be familiar with, might be serotonin, which is responsible for feelings of happiness. Dopamine, which is more of like your reward centers and kind of give you more happiness as well. Okay, it also gives kind of addictive behaviors to another degree. GABA, which typically has more of an inhibitory effect on different types of impulses. norepinephrine, endorphins, nitric oxide. Those are all different types of neurotransmitters, and each of those neurotransmitters has a different effect. Okay? And so what's kind of interesting is that a neurotransmitter might have an excitatory effect at one place where it might stimulate some kind of signal at one synapse. But if you take that same neurotransmitter to a different synapse, it might have a completely different effect. It might have an inhibitory effect. Okay? So an example of that we saw already was acetylcholine. Okay? So acetylcholine in neurons and as well as in muscles will have an excitatory effect to stimulate a signal to be moved from one neuron to the next cell. Okay? But if you take acetylcholine into the heart, for example, into the cardiac muscle, okay, acetylcholine has an inhibitory effect on cardiac muscle, causing it to not contract as fast. It reduces heart rate. Okay? So different neurotransmitters can have different effects at different regions of the body. So it always kind of depends on where exactly you're looking. Okay. So one particular example is with depression. So depression is a condition that affects a lot of people, not just in our own communities, but across the world too. And so depression, kind of from a more clinical perspective here, is a result of an imbalance of neurotransmitters, namely serotonin, okay, as well as norepinephrine and dopamine in the brain. Okay. And so what happens in people who have depression is that they are not producing, and so actually one of the two things is happening. Either A, they're not producing enough serotonin, okay, which gives them that happy feeling. Okay. If you're not producing as much of it, you're not going to be as happy as often. Okay. Or they're reuptaking it too quickly. Okay? So serotonin is one of those neurotransmitters that gets removed through reuptake. Okay? And so if you're reuptaking serotonin too quickly, okay, it's not spending enough time bound to receptors in order to trigger that happy effect. Okay? So there is a reason why, kind of the more anatomical reason why people develop depression. Okay? So in order to kind of combat that, what physicians will do is they'll prescribe an SSRI, which you've probably heard of in these different depression commercials or infomercials for different medications. Okay. SSRIs stand for selective serotonin reuptake inhibitors. Basically, what these medications will do is they'll block the reuptake transport protein. Okay. So if you cannot reuptake any more serotonin, serotonin spends more time in the synaptic cleft for a longer period of time. Okay. If it stays in there for a longer period of time, it stays bound to receptors for a longer period of time. Okay. If it stays active with the receptors for a longer period of time, that is what gives you that more improved mood that antidepressants will actually, help individuals with. Okay. So it helps to give people the more happy feeling when they're dealing with depression. Any questions on depression and SSRIs here? Okay, cool. All right, so now when we're taking a look at neurons, neurons here are going to come in all kinds of different shapes and sizes. When we look at the cell bodies of them, some of them can range from a cell body of having about 5 micrometers in diameter to as much as almost 6 inches across. So depending on the neuron that you're looking at, you can have some very small neurons and some extremely large neurons as well. And then they're going to have various types of branching patterns, which are going to allow them to perform different types of functions. And in some cases, you can even have neurons that don't have an axon at all. You can have some neurons that have very long axons, some that can range anywhere from multiple feet long. And then some axons can be very, very short. Okay, it all just kind of depends on the particular neuron you're looking at. And we'll take a look at a few of those different types of axons over the course of these next couple weeks. Okay. So the first type of neuron we're going to be seeing here are called multipolar neurons. These make up the vast majority of the neurons inside the brain and the spinal cord. So multipolar neurons are neurons that will have multiple poles here. So they'll have multiple dendrites and then a single axon coming off. So these will typically be found within the central nervous system. Okay. The second type of neuron are called bipolar neurons. Okay. And so bipolar neurons are going to have one main dendrite and then one main axon. So they're going to have 2 major poles, which is kind of where it gets its name from. Okay. So you have one major dendrite and then one major axon. And so these will typically be found inside the ear, the receptors in the ear, the eyes, and then the olfactory areas of the brain that are responsible for sensing smell. Okay. And then the last type of neuron are the unipolar neurons. So unipolar neurons have a single pole. They almost look like a guy, sticking a flag into the ground. And so these have two branches here that are both going to act as a single axon. Okay. So here you have your dendrites. Okay. Then you have your one long axon all the way across here. And then you end up with a central process. And this central process will extend into the central nervous system. Okay. So we have multipolar cells, bipolar neurons, and then unipolar neurons as well. Okay. In addition, you have some other kind of funky looking neurons here. So this first one is called a Purkinje cell, which almost has like dendrites that look like a tree or branches of a tree, and then one single long axon with a bunch of axon terminals at the end. So this is what's called a Purkinje cell. And then we also have pyramidal cells, which have more of a triangular cell body with lots of dendrites all the way across the cell, and then again a single long axon coming off of one side of it. So like I said, they come in all kinds of different shapes and sizes, and they all perform very different functions. Okay. So those are all the neurons. Are there any questions on neurons before we go forward here? Okay. All right, so if there's no questions, we'll go ahead and move on to the neuroglia. Okay. And so the neuroglia are also called glial cells or glia. These are cells, like I said, that are going to vastly outnumber the neurons. Okay. And so they actually make up about half of the volume of the central nervous system, even though they're a lot smaller than the neurons themselves. Okay. Now, again, like I said, their job is not to generate any action potentials. Okay. Their job is not to provide any communication from the neurons, between the neurons, or to neurons, or from the neuron to the body. Okay. They're not generating any action potentials here. But they do have the ability to divide and multiply while neurons do not have that ability. Okay. So when we look at neuroglia, we have 6 different types of neuroglial cells. Okay. The first 4 are going to be found inside the central nervous system, and these are the astrocytes, the oligodendrocytes, the microglia, and the ependymal cells. Okay. So like I said, those 4 are only found in the central nervous system. Okay. The other 2 are the Schwann cells and the satellite cells, and those are only found inside the peripheral nervous system. Okay. So we're going to take a look at those 6 different types, and we're going to start by taking a look at the astrocytes. Okay. So the astrocytes are going to be the largest and the most numerous of those 6 neuroglial cells. Okay. So astrocytes almost look like they are like these star-shaped cells. And they're going to have all these different types of arm-like processes branching off of its own body here. Okay, so here we see all these different arm-like processes, and we'll see specifically where these guys become very important next week when we look at the brain. Okay. And so these processes are going to be able to make contact with blood vessels here, and so this will help to form the blood-brain barrier. Okay. These processes can also help to form contacts with the neurons. Okay. And that'll help to support and hold up the neurons in place. And then they can also hold up or make contact with the pia mater, which is a part of the part of protective membrane for the brain and the spinal cord. Okay. So these astrocytes are going to be probably one of the more important ones because they're going to be performing multiple different types of functions. Okay. The first function is going to be to provide the strength in order to support and hold all the neurons in place. Okay. So the neurons that kind of are present are not going to be kind of floating around and moving from point A to point B. Okay. They're going to be held in place by all of these astrocytes, and they're going to kind of secure them. Okay. The second function is going to be to form the blood-brain barrier. Okay. So when the processes of those astrocytes make contact with the capillaries inside the brain and the spinal cord, specifically inside the brain here. Okay. That is what's going to form the blood-brain barrier. Okay. And that is going to be able to regulate the astrocytes specifically, as well as the endothelial cells of the capillaries themselves. They're going to be able to regulate what substances are able to cross from the blood into the brain and vice versa, and what can't. Okay. So this is a way for us to be able to regulate what comes into the brain and what doesn't. Okay, so that's a very, very sensitive structure, a very important structure to help protect the brain. Okay. The third function is going to be inside the embryo, and so the astrocytes are going to produce these various types of chemicals that are going to help to allow the neurons to be able to grow, move from point A to point B, and then also form synapses within the brain. Okay, so that's function number 3. Okay, number 4 is it's going to be, and number 4 is going to be one of the more important functions. Excuse me, is it's going to be able to perform or maintain the environment, the chemical environment of those axons, okay, of those neurons here. Okay. It's going to make sure that the chemical environment around all those neurons is appropriate in order to help allow action potentials to take place. Okay, so it's going to make sure that there's enough sodium in order to help generate action potentials. It's going to make sure that there's enough potassium and enough chloride available to help, either generate action potentials or have enough to stop action potentials if need be. Okay. So they're going to be able to regulate the chemical environment surrounding various types of neurons. Okay. And then the last function is it's going to help with learning and memory. Okay, so it's going to be able to make sure that different neural synapses can form, okay, between different neurons over the course of your lifespan. Okay. And as you form different neural synapses and different neural contacts, that is what's going to allow you to be able to learn, make connections between different material that you might have learned, and also be able to help you kind of solidify memory of different events or different things you've learned and things like that. Okay. So astrocytes do all kinds of different types of functions throughout the nervous system here. Okay. The second type of neuroglia are the oligodendrocytes. Okay. And so the oligodendrocytes are these cells that are going to have these processes here. Okay. And all these processes are going to branch off and then latch onto and surround axons, okay, and cover the axons. Okay. Each of these processes here is going to form what's called the myelin sheath, which is a lipid, kind of more fatty covering, and it has a little bit of protein as well. Okay. And what that myelin sheath will do is it's going to help increase the speed of those action potentials. Okay. So it allows action potentials to move a lot faster from the neuron or from the axon hillock down to the axon terminal. Okay. So the goal is to insulate the axon and then increase the speed of the action potential. And we'll look at how that's able to take place later. Okay. Okay. Those are the oligodendrocytes. Okay. The third type of the third type of neuroglia are the microglial cells, okay, or microglia. Okay. And what they do is they essentially will act as the white blood cells of the nervous system. Okay, so someone remind me, what do white blood cells do?
They fight bacteria.
Anyone
They
remember?
They act as macrophages, remove debris.
Right. Exactly. So they're going to be fighting off different bacteria, fighting off debris. They're going to be kind of the defenders of the body, right? So
Yes.
microglia are going to basically be doing the same thing, but specifically in the brain. Okay. So if there's any cellular debris that ends up in the brain, if there's any neurons that end up getting destroyed, or any kind of other substance that shouldn't be in the brain, or any bacteria, whatever it may be, okay, microbes, any damaged tissue, the microglia are responsible for phagocytizing it, engulfing it, destroying it, and then eliminating that from the picture, essentially. Okay. So here, for example, here we have a neuron. Okay, this neuron is going to undergo apoptosis. It's going to end up getting destroyed. And so instead of keeping that dead neuron present inside the brain, what the microglia will do is it's going to engulf it. Engulf it, destroy it, phagocytize it, and then now it's no longer going to be impacting the brain. Okay. So this is especially going to be important when it comes to any damaged nervous tissue. Okay. And so, for example, football players who go through large periods of time where they're having lots of physical head-to-head contact can sometimes have some damage inside the brain, where they end up with CTE or a lot of concussions, where some of the neurons get damaged. Okay. So the microglia that come in that kind of clean up that damage, okay, or just phagocytize all those dead neurons, okay, that's going to be taken care of by the microglia here. Okay, they're going to engulf and destroy all of those damaged cells, okay, and then again remove them from the picture. Okay. So because they kind of act as white blood cells, they're actually going to develop and kind of originate inside the red bone marrow, just like any other blood cell would. Okay? And then as it starts to develop, they eventually migrate their way into the central nervous system, where they will eventually start to just reside and live. Okay. And the last type of neuroglia that we find in the CNS is called the ependymal cells. Okay? And so the ependymal cells are cells that will line the ventricles of the brain, okay? And the central canal of the spinal cord. We'll look at exactly what we mean by that when we look at the spinal cord on Wednesday and then the brain next week. Okay. But there's going to be specific regions of the brain and the spinal cord that are going to be lined by ependymal cells. Okay. So these ependymal cells are responsible for producing, monitoring, and then circulating the cerebrospinal fluid, or your CSF. And so I will look at what the CSF does specifically later this week and then next week as well. But this is what's going to help. These particular cells are going to help to form the blood cerebrospinal fluid barrier in the brain. Okay? So they're going to be able to regulate what materials are able to pass from the brain or from the cerebrospinal fluid and into the blood and vice versa. Okay, so those are the 4 types of neuroglia that we find inside the CNS, the central nervous system. So we're going to have the astrocytes, the microglia, oligodendrocytes, and then the ependymal cells. Okay. In the peripheral nervous system, we're going to have only 2 types of neuroglia present, and so these would be the Schwann cells and the satellite cells. Okay. These Schwann cells are going to be individual cells that will wrap themselves around a single axon, okay, and they're going to produce myelin sheaths, okay, just like the oligodendrocytes do. So Schwann cells will produce myelin sheaths around a single axon, and that is going to help the action potentials move a lot faster. Okay. Now, these also have a slight ability in allowing axons to regenerate themselves, assuming there's not significant damage present. Okay. So that's going to be these guys here. Okay. Schwann cells producing myelin sheaths in the peripheral nervous system, whereas oligodendrocytes will produce the myelin sheaths in the central nervous system. And they also look a little bit different too as well. Okay. Then the last type of cell are called the satellite cells. Satellite cells are these kind of more flat cells that will cover the cell bodies of the peripheral nervous system. And so those are what we refer to as ganglia. So ganglia are these collections or these clusters of neuronal cell bodies outside of the central nervous system. Okay. So satellite cells are going to be covering the cell bodies of all of these ganglia inside the peripheral nervous system. And what they'll be doing is they're going to be helping to provide some structural support, and they're also going to be regulating what materials and ions and things like that are able to pass from the interstitial fluid into the cell body. Okay. Okay. So they're going to be able to regulate what comes in and out of that cell body. All right, so those are the 6 neuroglial cells here. Okay. So before we go forward here, are there any questions on the neuroglia before we go forward here?
No, I think it's clear.
Okay. Okay, cool. All right, so this is just a quick little picture here. So here we see some Schwann cells covering an axon. So here's an axon here. Here we have a Schwann cell. You can see its nucleus. And then you're going to see this yellow structure around the axon. Okay. That is the myelin sheath. Okay, so this is the lipid covering that helps to allow those action potentials to move faster. Okay. Now, not all Schwann cells are going to be producing myelin sheaths around their axons. Okay. So for example, here we have a Schwann cell, here we have a bunch of axons, but we don't see any myelin sheaths covering them. Okay. So these would be what we refer to as unmyelinated axons. Okay. So some Schwann cells will produce myelin sheaths, other ones won't. It just kind of depends on where exactly that particular individual is in development or where in the body you're looking. Okay, so this is kind of reiterating what I just mentioned here. So axons that are surrounded by a myelin sheath are what we refer to as myelinated. Okay, so in order for myelination to take place, what we're going to see is our Schwann cell is going to surround an axon. Okay, it's going to kind of coat it, okay, cover all the way around, and then start producing myelin, okay, until it gets completely insulated by that myelin sheath. Okay, so that's going to increase the speed of an action potential. Okay. Now, what's going to happen is you're going to have a region of your neuron, okay, and each neuron is going, or each region of a neuron is going to be surrounded by a myelin sheath. Okay, then there's going to be a little bit of space, and then another Schwann cell producing a myelin sheath. Okay, then a space, and then another one. Okay, so each of those gaps in the myelin sheath is what we call a node of Ranvier. Okay, so this is what we find over here. Okay, so this would be your Schwann cell producing a myelin sheath. Here we have a region where we don't have any myelin. Okay, we just have the axon. So this would be a node of Ranvier in between this Schwann cell and then the next one producing its next myelin sheath. Okay, so the gaps in the myelin sheath are the nodes of Ranvier. Okay. Now, any axons that do not have a myelin sheath are going to be considered unmyelinated axons. Okay, so those ones do not have action potentials that move nearly as fast as myelinated axons. Okay. Now, the amount of myelin that we develop is going to end up increasing from birth to maturity. So from the time you're born, you don't really have that many myelinated axons. Okay. And so that's part of the reason why if you're looking at a baby who's like first learning how to walk and they're a little bit clumsy, they might trip over themselves. And the reason for that is because most of their myelin in their peripheral nervous system has not developed yet. And so they're starting to become more uncoordinated. Okay. So next time you see kind of a baby trip, you just be like, oh, you know, your myelination hasn't completed just yet. Okay. That's essentially what's going on there. Okay. And then eventually, as you get older, you end up producing much more myelin, so you can actually perform lots of more coordinated motor movements. Okay. And so again, there's going to be two neuroglia that are responsible for producing the myelin sheets in Schwann cells. In the peripheral nervous system, myelin will be produced by the Schwann cells. In the central nervous system, that myelin gets produced by the oligodendrocytes. Okay. So one way that I kind of use to kind of help myself keep that straight is that the O and the C, they look very similar, okay, very similar looking letters. So those two are pairs. Okay, so oligodendrocytes always in the CNS, Schwann cells are always in the PNS.
So the amount of myelinated
So,
sheets doesn't it
yeah.
also decrease with age, like when you get really old?
Yeah.
Don't you lose
to some
myelination?
degree, yes. Yeah.
Okay.
Yeah. So to some degree, oh yeah, go ahead. Yeah. So as you get older, your Schwann cells, you start to lose a little bit more of your myelin. Not a lot of it, but in certain regions, you might not have as much myelination as you once did, and so you end up getting more of like the tremors and things like that as you get older. But it's not significant enough to be like a huge downfall to like your nervous function.
Oh, okay. So it doesn't like revert back to when you were like a baby, right? It doesn't revert, like, you know.
No, it never reverts all the way back.
Oh, okay.
No.
Okay.
Yeah.
Well,
Yeah, it'll just a little bit, but not. Yeah, just a little bit, but not all the way. Yeah,
For sure.
just a tad.
Okay.
Okay. So this is what myelination would, or what action potential speeds would look like if you're looking at an unmyelinated axon versus myelinated axons here. Okay. And so here we have a neuron that's unmyelinated. So we see that after 1 millisecond, it's gone this far. After 5 milliseconds, your action potential is now this point. And then after 10 milliseconds, the action potential has now moved to this particular region. Okay. So it's moving relatively slow over this period of time. The reason for that is because you have to have all these sodium channels to create those action potentials. Problem is sometimes these sodium channels can be a little bit leaky. And so if sodium comes in, sometimes it might leak out a little bit. And so that impacts the overall voltage inside your axons. Okay. So if you have sodium coming in and leaking out, sodium coming in, leaking out, coming in, leaking out, okay, you're not going to be able to move the action potential as far as it should, or as far as you would like it to move. Okay. It's going to be moving relatively slow. Okay. Now, in the case, and you have to have these sodium channels all the way throughout your entire axon. Okay. In the case of myelinated axons, you're only going to have these sodium channels inside the nodes of Ranvier. Okay, so in between your myelin. Where you have myelin sheets, you will not have any sodium channels. Okay, and so for example, here we're going to have some sodium come in. Okay, once it comes in, it's going to be able to just jump straight to the next node because there's not going to be any leakage at any point until you reach this particular region here. Once the action potential reaches this region, sodium can then flow in, and then boom, you jump to the next node. Okay? And then boom, you jump to the next node. Okay? So that's what we call saltatory conduction, okay, where action potentials seem to jump from one node to the next. Okay? And so as a result, they end up moving a lot faster down the length of your axon than they do if you had an unmyelinated axon. Okay? Does that make sense?
Mm-hmm.
Yeah. So with myelination, action potentials are able to basically jump from one node of Ranvier to the next node. Okay. And so this is kind of a more in-depth picture of what this would look like here. Okay, so here we have some myelin, more myelin here. Okay. In between each region, you have the myelin sheet, sorry, the node of Ranvier. Okay. So inside the node of Ranvier, you're gonna have your sodium channels, which are going to allow your voltage to increase. You're gonna have an action potential occur there. Once you have an action potential, it just boom, jumps straight through the myelin, okay, and then moves to the next node. Okay, then you add more sodium in, and then boom, jumps to the next node, and so on and so forth. Okay? So action potentials are able to move a lot faster using myelinated axons than they are through unmyelinated axons. Okay, this is what it would look like if you were to take a look histologically. So here we see a neuron. We see some myelination surrounding this particular neuron, okay, this myelin sheath, this very dark region here. Okay. Versus unmyelinated axons, here this would be one axon, another one here. Okay, but you can see there's not nearly as much myelin around. Okay, there's no myelin surrounding any of these axons. Okay. All right, so question here. So in the figure shown, how is this neuron classified structurally? Okay, is this a multipolar, pyramidal cell, bipolar neuron, Purkinje cell, or a unipolar neuron?
Bipolar, right?
Unipolar?
It's not bipolar.
Not bipolar?
This would be unipolar. Right, so unipolar, you have a single pole coming straight off of your cell body. Okay. If we were looking at a bipolar neuron, it would look something like this. So you have a dendrite on one end and then a axon on the
That
other.
of the soma. Okay.
But here we're looking at a unipolar cell which has just one single pole. Yeah.
Okay, got it.
How about
Okay,
the multi?
Sorry, what was that?
So what about the multipolar? How will they look? It has two dendrites, right?
So multipolar, no, it would have more than one dendrite. So if we were looking at a multipolar, here we'd find, let's say this is our cell body. Okay, it's going to have a bunch of dendrites coming off on one end. Okay, lots of branching off our dendrites here. Okay, and then one single axon. So a multipolar would have multiple dendrites on one side and then one single axon. So this would be, I'll just put multipolar here. If we're looking at bipolar, we would have a cell body with one single neuron, or sorry, one single dendrite, and then one single axon.
Mm-hmm.
Cool.
Mm-hmm.
So,
All right. Any other questions here? Okay. All right. So let me go and clear all this. Okay. Okay, next question. So the myelin sheath of the central nervous system neurons is produced by which cells? The ependymal cells, Schwann cells, oligodendrocytes, astrocytes, or microglia?
Oligodendrocytes.
astrocytes?
So I'm hearing oligodendrocytes, I hear astrocytes. What else
Make
do you
it
think?
fine, guys. Come on.
See. Pretty sure.
Anyone have an idea?
Like
So
a C.
the answer,
Let's see.
yeah, so the answer is C, correct, the oligodendrocytes. So oligodendrocytes will be producing myelin sheaths in the central nervous system. Astrocytes do a bunch of other things in terms of kind of the metabolism of the neurons.
Mm-hmm.
Which ones would be producing myelin sheets in the in the peripheral nervous system?
Schwann cells.
Oh, that's what I was thinking too. Yeah, I was like, I don't know. I was like, maybe Schwann cells, but I don't think so.
Yeah. Yeah. So oligodendrocytes produce it in the central nervous system. Schwann cells produce myelin in the peripheral nervous system.
Mhm.
Cool. All right, so that's that. Actually, we'll kind of go back here. What's the role of the microglia? What do microglia do?
They support
Anyone remember?
the nervous system, correct? I mean the nerves, the neurons. Provide nourishment and support.
But how? What do they do? Not nourishment. Those are the astrocytes.
Phagocytes production.
Yes.
Yes.
So the microglia are responsible for phagocytizing any bacteria, microbes, debris, damaged neurons, anything that gets destroyed or anything that shouldn't be in the brain. The microglia are going to be the ones that clean all that up. Yeah, they're the white blood cells of the brain.
They're like the white blood cells of the brain.
Exactly.
All right.
Nice.
Exactly.
Later.
Beautiful. And then last thing, the ependymal cells. Anyone remember what the ependymal cells produce?
Mhm.
It was a very big word.
Let's go back in the notes.
Let's
Ependymal cells. Oh.
ependymal cells.
You remember?
Not the top
So
of my
the
head.
ependymal cells, they'll be producing the cerebrospinal fluid.
Oh.
Okay.
Yeah, so kind of go back. Let me kind of go back here. Yeah, so they're going to be responsible for producing the cerebrospinal fluid.
CSF.
And again, we'll look at
Oh.
yeah. So we'll look at its function later on in the this week and the next week too.
Okay.
Good. Okay. Next question. So gaps in the myelin sheath are called the what? Nodes around VA, trigger node or trigger zone, neuromuscular junction, axon hillock, axon terminal.
Hey, nodes of Ranvier.
What do we think?
Lock it.
Yes, the nodes of Ron VA. Perfect. Perfect. Good. All right, cool. Okay. Any questions on the neuroglia before we go forward here?
Not yet.
Okay.
Okay.
Cool. All right. So getting towards the end of this. So when we look at the nervous system, so we're kind of go back from looking at things at the micro level. We're going to kind of look at the macro level for a bit here. Okay. When we look at the nervous system, whether we're looking at the brain or we're looking at the spinal cord, we're going to find different regions that appear more white and regions that look more gray. Okay. So this is a brain that's been dissected. Okay. And so you can see this innermost region is going to be mostly white, while the outermost region is going to be more kind of grayish, brownish color. Okay. So each of those different regions is going to have different substances in it. Okay. So the white region we refer to as the white matter. Okay. So the white matter here is going to be made up of mostly myelinated axons. Okay. So the myelin sheets are made up of lipids. So this is going to be mostly fat. Okay. And so the myelin actually gives these particular regions their white color. So whenever we think of white matter, we think there's going to be lots of myelin present there. Okay? To a lesser degree, there's also going to be a few unmyelinated axons, but for the most part, there's going to be a lot of myelinated axons present here in the white matter. Okay? Gray matter appears more gray because there's no lipids there. There's no myelin. Okay? So in the gray matter, we'll just find the neuronal cell bodies. Okay? We'll find dendrites. We'll find unmyelinated axons. We'll find axon terminals, and then we'll find lots of neuroglia as well. Okay, but we're not going to find anything that's myelinated inside the gray matter. Okay, so the only place we'll see myelination is going to be inside the white matter. Cool. Okay, that's that. Okay, so a question here. So in the figure shown, what gives the indicated region its distinctive color? Okay, so here, we're looking at this particular region over here. This is going to be the axon terminals, dendrites, myelinated axons, neuronal cell bodies, or unmyelinated axons.
Myelinated axons.
Right, myelinated axons. They're going to give the white color for the white matter. Perfect. Oops, perfect. Okay. Now, the myelin can be targeted for specific autoimmune diseases. Okay? And so one of the major autoimmune diseases that happens in the nervous system is called multiple sclerosis, or MS. Okay? And so what happens in people who have multiple sclerosis is the myelin sheets around the central nervous system end up getting destroyed. Okay? So this is what your myelin sheets should look like. They should look like nice and whole, nice and full, nice and, nice and healthy, right? Okay. But in the case of someone who has multiple sclerosis, they start producing antibodies against their own myelin sheets. Okay? And they start breaking down their own myelin. Okay? So if they start breaking down their own myelin, okay, what effect do we expect to have that on the action potentials? Our action potential is going to be moving faster or moving slower?
Slower.
They're right. They're going to be moving slower. Okay? So if we start to break down our myelin sheets here, our action potentials can move slower. And if it gets severe enough, you may actually short circuit the entire process altogether and stop action potentials at all. Okay? And so for people who have multiple sclerosis, sometimes they have a very difficult time with like lifting up their muscles because the action potentials don't move to their muscles effectively, and sometimes the action potentials may cut off before they even reach the muscles. Sometimes they get abnormal sensations because, again, the myelin sheets are not transmitting those different sensory signals or motor signals as they should. Sometimes they develop double vision. Okay, it results in a whole lot of different problems, all because you destroyed the myelin sheets, and so you're not able to transmit action potentials as effectively. So this typically appears between the ages of 20 and 40, and typically in females more often than in males for unknown reasons. But one way that is people are able to kind of diagnose it is by using an MRI. Okay? And so when you put someone's brain under an MRI, it should look pretty much like this. Okay? It should look pretty full and pretty much gray all the way across the board. Okay? But in someone who has multiple sclerosis, they'll develop these white plaques that will be visible under this MRI. Okay? And so these plaques are where you're going to start to see some demyelination take place. Okay? So we have plaques developing inside the white matter, inside the brain and the spinal cord, which causes a lot of these problems, across the brain and across the body.
Crazy.
So that is, yeah.
Is it some type of cancer?
No, it's not a cancer. It's just an autoimmune disease. So for some reason, certain people's bodies will start producing antibodies against their own cells or against their own things that their cells will be producing. And so they just produce extra antibodies when they shouldn't be. And those antibodies specifically target their own nervous system. Yeah, cool. Any other questions here? Alrighty, so that's multiple sclerosis. Okay, so last couple things here. So here we're looking at neural circuits. And so the central nervous system is going to be made up, like I said, of 85 billion different types, or 85 billion different neurons. And each of those neurons is going to be a part of different circuits. And so these circuits can look different depending on the type of function that circuit needs to be able to perform. And so we looked at the first circuit, which is called a simple series circuit. So in a simple series circuit, all we have is just a single presynaptic neuron synapsing with a single postsynaptic neuron. Okay? And that's it. Okay? You just have one single neuron communicating with one single other neuron. Okay? That's the most simplest of the different types of circuits, but it gets a lot more complicated from there. Okay? The first one the next one that we're going to see is called a diverging circuit. Okay? So in a diverging circuit, you have one presynaptic neuron that's going to synapse with multiple postsynaptic neurons. Okay? And then from there, each of those neurons will then synapse with other neurons and so on and so forth. Okay? And so you're starting with one single neuron and then ultimately stimulating multiple other neurons in the chain. Okay? And so this is useful to help amplify a particular signal. Okay? Amplify a signal that you want the brain to be able to communicate to some other region. Okay? Or a neuron to amplify to another region. Okay, that's a diverging circuit. The second type of circuit we can see is called a converging circuit. Okay? And so in a converging circuit, we have multiple neurons all converging, multiple presynaptic neurons all converging onto a single postsynaptic neuron. Okay? And so this allows us to be able to, this is typically going to be found in muscle fibers. And so muscle fibers, especially one thing that you'll learn if you take physiology, is that muscle fibers actually get stimulated by multiple neurons. And so what will happen is you'll have multiple motor neurons all stimulating a single muscle fiber to help create a stronger muscle contraction. Okay. So that would be an example of a converging circuit. Okay. The next one is called a reverberating circuit. So in a reverberating circuit, you have one neuron that stimulates another one, stimulates another neuron, and that stimulates a final neuron in the chain. Okay. This final neuron here will then send impulses back to one of the previous neurons in the chain here. Okay. So it kind of reverberates back to one of the previous neurons. And so this is really important for lots of behaviors that involve patterns. Okay. So like breathing or rhythmic movements, for example, okay, or rhythmic functions. So breathing, for example, is going to be performed by lots of reverberating circuits, coordinated muscle movements, sleeping, short-term memory. Okay, these all involve reverberating circuits where you need a postsynaptic neuron to eventually reverberate back to and communicate with a previous neuron in its own circuit. Okay. And then the last one is called a parallel after discharge circuit. So in a parallel after discharge circuit, you have a single presynaptic neuron, okay, that will stimulate a group of other postsynaptic neurons. Okay, and each of these other postsynaptic neurons will then stimulate, okay, and synapse with a single final neuron here. Okay. So you have one starter, okay, that's going to stimulate multiple other neurons, and then those other neurons are eventually going to synapse onto a single postsynaptic neuron. Okay. And so this is important for any functions that allow you, or any functions that require you to pull information from multiple places. Okay. So precise activities like doing mathematical calculations, okay, they require you to be able to pull from different types of areas, okay, different types of things that you've learned, okay, and then eventually synapse back onto a single neuron to get you your final answer or help you do the final task, whatever it may be. Okay. So those are the 5 different types of neural circuits. Last thing here is with regeneration and neurogenesis. Okay, so throughout the course of our lives, our neurons and our nervous systems are going to be able to grow, change, and then remap itself. Okay, so that's what we call the ability called plasticity. Okay, so plasticity means that your nervous system is able to change over time, it's able to grow over time, and it's able to kind of remap and form new synaptic contacts with other neurons. Okay. So this is going to be done by either forming new dendrites, providing new proteins, or again, providing new synaptic connections between one neuron and another neuron that wasn't previously there. Okay. So plasticity is going to be really important, especially when it comes to learning. Okay. One thing that is kind of important here is that mammalian neurons, so neurons like humans, squirrels, things like that, other mammals, our neurons are not very good at regenerating. Okay? In the peripheral nervous system, there is a little bit of regeneration that can take place, but this can only happen if the dendrites are undamaged, or if the cell body remains intact, okay? Or if the Schwann cells around them are also remaining intact as well, and active. Okay? So the dendrites and any myelinated axons can be repaired if the cell body and the Schwann cells present are able to still work. Okay. If that's not the case, they're not going to be able to regenerate. Okay. In the central nervous system, there's pretty much no ability to regenerate. Okay. And the reason for that is because the oligodendrocytes pretty much don't allow that to happen. And then astrocytes also kind of serve kind of like an overprotective role. And so if there is kind of any damage inside the central nervous system, the astrocytes will compensate for that by producing lots of scar tissue inside the brain. But that scar tissue like acts as a kind of physically gets in the way of regeneration. And so regeneration cannot happen in the central nervous system, specifically because the astrocytes kind of produce that scar tissue that gets in the way, and then oligodendrocytes don't allow that to happen either. And there's also an absence of any growth stimulating cues. So there's not really a whole lot of growth that can take place within those neurons. However, even though we cannot regenerate the neurons that we do have, it is possible to produce new neurons through a process called neurogenesis. Okay? And so neurogenesis only happens in specific areas of the brain, namely the hippocampus, which is involved in learning. We'll look at that in more detail next week. But that's going to be one place where we can actually produce new neurons, which is important because obviously you'd want to produce new neurons when you're learning new things. Okay. And that is it for today. Any questions